All right, ladies and gentlemen, this video will help you complete your notes from January the 11th. This section of the video is picking up halfway down the second page. We're under letter F where it says diploid. Okay, so here we go. So a cell that contains both set of homologous chromosomes is said to be diploid. Remember, a diploid means that there are two kinds of chromosomes in each cell, and the number for chromosomes in a diploid cell is represented as 2n. The diploid cells are the somatic cells. The diploid cells are the body cells. The diploid cells are the ones that we are going to produce during mitosis, and these are used primarily for growth. These cells are primarily used for repair and healing. So taking a look at an example, in a fruit fly, 2n equals 8, okay? What this means is that n is the different types of chromosomes that the organism has. In humans, 2n is 46. That means that we have 23 types of chromosomes. We have chromosome number 1, chromosome number 2, chromosome number 3, et cetera, and so forth. Human cells contain 23 pairs of homologous chromosomes. That means diploid cell contains 2 complete sets of chromosomes. You get 1 set from mom, you get 1 set from dad. We are at number 6. Mitosis, however, is going to produce identical daughter cells. So during cell division of a diploid cell, during cell division of a body cell, during cell division of a somatic cell, which is mitosis, again, growth and repair, healing. The mother cell is diploid, and we end up with 2 identical diploid daughter cells. So we are literally just reproducing the cell in and of itself. However, when it comes to gametes, that's not going to be the case. Gamete cells, which are used for sexual reproduction, only have a single set of chromosomes. This is letter G on your notepad, on your note catcher. Chromosomes, again, are going just a single set in your gametes. Gametes are what we call haploid cells. Again, sometimes we call them haploid cells. We can call them gametes. We can call them sex cells. This all works to describe a haploid cell. The number of chromosomes in a haploid cell is half the number of chromosomes that would happen in a normal diploid cell. So the number of chromosomes is 1n. When fertilization occurs, we get half from mom, we get half from dad, and those come together and make a whole diploid number inside the zygote. So again, we are looking at chromosomes in gametes. Gametes are your sex cells. Gametes are your haploid cells. In a haploid cell, you only have a single set of chromosomes or half the number of chromosomes. Again, and the reason for this is so when we have fertilization, the zygote will have the correct number of diploid cells. And again, I mean, it's really simple. Like 1n in the egg, 1n in the sperm equals 2n in the zygote. And 2n brings us back to that diploid number. We are moving on to number 4. The cells that produce eggs and the cells that produce sperm are diploid. So how do you get the egg and sperm to get to be one end? How do you get those diploid cells to turn into haploid cells? The answer to that is meiosis. So remember, mitosis is for making diploid cells. Meiosis is for making haploid cells. I know it's really similar, but again, I always remember it's because meiosis makes me. Meiosis makes me. And what made me are my parents, which means that there had to be some gametes involved. All right. So we are looking at phases of I'm sorry, we're not there yet. I apologize. This is number 5. Meiosis is a process that we call reduction division. So we reduce the number of chromosomes in a cell, and then we divide up those cells. So in other words, we reduce the chromosomes for each cell by half. So a human cell would start with 46, end with 23. If it was a fruit fly, it would start with 8 and it would end with 4. If it was a dog, it would start with 78 and it would end with 39. Sure, that sounds about right in terms of my math. We reduce it by half through the separation of the homologous chromosomes. We separate those matchy homologous chromosomes. All right, we are now on to number 3, phases of meiosis, excuse me. And I believe this is going to take us through A through D. Okay, so meiosis only occurs in the production of egg and sperm cells. So meiosis only is happening when we are creating a gamete, when we are creating a sex cell, when we are creating a haploid cell. The purpose of meiosis is to reduce the chromosome number in the egg and or the sperm by half, because again, when we are reproducing sexually, the idea is the offspring is going to get half of its chromosomes from mom and the other half from dad. Now, in meiosis, unlike mitosis, it is meiosis, exactly like mitosis. Before any of this begins, we have to replicate our chromosomes. But remember, this is reduction division. The idea is that we want to reduce and divide. So actually, there's 2 divisions that happen here, and we'll get into that in a moment. So we say there is meiosis 1 and meiosis 2. In meiosis, one diploid cell ends up producing 4 haploid cells. And those haploid cells, again, they're gametes, they're sex cells, and we'll talk about those a little bit more in a moment. But again, the purpose here of meiosis is to produce the gametes, is to produce the haploids, is to produce the sex cells. That is the entire purpose of meiosis. All right, so you have this diagram in your notes already. You can read through it as need be. All right, but you'll notice we have meiosis 1, and then we have meiosis 2. Meiosis 1, we end with 2 cells. By the end of meiosis 2, we end up with 4 daughter cells. All right, we are moving on to interphase. This is the top of page 4, number 1. So prior to meiosis 1, all of your chromosomes have to be replicated. This is always happening in interphase. It doesn't matter if it's meiosis or mitosis, it's what they have in common. We are always going to replicate our DNA first because, again, the idea is at the end of cell division, you want a cell that does something. In order for a cell to do anything, it has to have some DNA in it. And in order for it to have DNA, that DNA has to be replicated during interphase. When it replicates its DNA, you are going to end up with identical sister chromatids, again, held together by a centromere. We've seen this before. It is exactly what happens during interphase for mitosis. So nothing special has happened yet. All right, to me, prophase 1, this is like the best thing ever. This is probably the most important phase of meiosis because this is, well, one of the most important. There we go. How's that? So during prophase, just like before, chromosomes shorten and thicken. Big surprise. Chromosomes pair up with its corresponding homologous chromosome to form a tetrad. Okay, so again, they pair up with their homologous chromosomes. That's nothing new. There are 4 chromatids in a tetrad. The pairing of homologous chromosomes does not occur during mitosis. All right, so there is that. So why is this so freaking important? Oh, no. Okay, they didn't talk about it. All right. I will say this here. I know it happens later on as well in the notes. The reason why prophase 1 is so important is this is where we have a process called crossing over. So my little tetrads are really close to each other, and it's almost like they're holding hands. And when they hold hands, they like swap genetic information. So this is one of the areas in meiosis. So meiosis 1, prophase 1, where we are going to have an increase in genetic variability, which is awesome because it's kind of the entire point of sexual reproduction is to create new and interesting combinations of genetics to help improve survivability. During metaphase 1 and anaphase 1, so we're moving down into numbers 3 and 4. During metaphase 1, the tetrads line up at the center of the cell. Anaphase 1, the tetrads break apart and they move to opposite sides of the cell. So metaphase, they're still lined up in the middle. You will see it like a double line for metaphase 1. Anaphase, they're still being pulled away. We remember this. And big surprise, the difference here, though, in anaphase 1 is that the sister chromatids are still attached at their centromeres. So that is a difference from mitosis. So FYI. All right. Next phase, of course, is telophase and cytokinesis. Again, this is very, very similar. At the end of telophase and cytokinesis, the cell separates into 2 cells. Meiosis 1 results with 2 haploid daughter cells. Okay. Each daughter cell has half the number of chromosomes as the original cell. Because remember, in metaphase, we separate those homologous chromosomes. They move apart from each other. That was what happened. So now we end up with half the number of chromosomes as the original cell. At the end of telophase 1, though, my sister chromatids are still going to be attached to each other. Kind of important. Okay. Now, there is no new interphase, all right? We immediately, from telophase 1, we immediately move into meiosis 2 or prophase 2, all right? Prophase 2 is kind of boring. Sister chromatids start again. They're there, and they're going to start moving towards the center. Literally nothing exciting happens during prophase 2, like at all. It's like boring. It's like watching paint dry. In metaphase 2, the pairs of sister chromatids line up at the center of the cell. This is going to be a single file line, this time with the centromere at the center and the sister chromatids on either side of the equator. This looks a lot like mitosis metaphase, except for we are now only dealing with half the number of chromosomes. Anaphase 2, big surprise, they move away from each other. They move towards opposite sides of the cell. Anaphase away, we remember that. Telophase and cytokinesis, again, for meiosis 2, we now have 2 separate nuclei, and the cytoplasm pinches off. And we now have a total of 4 haploid daughter cells. So, the importance of meiosis, all right? So, we are now at letter G, numbers 1 and 2. One diploid cell, one 2N cell, is divided in order to produce 4 1N cells. Okay, so one diploid produces 4 haploid cells. Again, number 2, importance of meiosis. The chromosome number of eggs and sperm are reduced by half. What this means, of course, then, is that the zygote, once fertilized, will have the correct number of chromosomes. Because again, we reduce them by half. So that way, when fertilization happens and they come back together, we have a proper number of chromosomes again. Now, during prophase 1, and I sort of talked about this already a little bit, hugely important. If you remember nothing else from meiosis, please remember this: crossing over. During prophase 1, as those pair of chromatids, they will line up next to their homologous pair. So they all get, kind of lined up and they hang out together. This produces a tetrad. Tetra as in 4, because I see 1, 2, 3, 4 chromatids. So while this is happening, because it's getting kind of crowded in there, these chromatids sometimes will twist around each other. And when they do that, certain portions will sort of break off and they attach to other chromatids. This is called crossing over. And what I want to point out is this last picture over here, where some of this purple chromosome has a little bit of orange now. Some of this orange chromosome has a little bit of purple. So they have swapped some areas of genetic information. This increases genetic variability, which again is the entire point of sexual reproduction. Because sexual reproduction, again, it's slow and you don't make very many offspring, but what you do get out of it is genetic variation, which is awesome because it helps with survival of a species if there's changes to the environment. We are now flipping over, I want to say, to page number 5. At the very, very top, crossing over is the exchange of genetic information or genes between segments of homologous chromosomes during meiosis. Again, this is happening during prophase 1. Prophase 1, probably one of the most important phases of meiosis when it comes to creating these sex cells, because again, it's creating genetic variability. The result is that the offspring are going to have new combinations of genetic information. So, for example, again, we see this blue chromosome lined up next to its homologous pair of the yellow chromosome, and they are now a tetrad. The tetrad gets crowded, they sort of twist around each other, and they exchange genetic information. By the time it's time for them to sort of separate as meiosis continues, we now have what we call a recombinant chromosome. And these chromosomes in the center are recombinant because they have been reconfigured. On the outside, notice how this one's still all 100% blue. This one on the outside is all still 100% yellow. They are non-recombinant chromosomes, okay? Anytime you have a new combination, we have a recombinant chromosome. This creates genetic variation, which is the whole point of sexual reproduction, to create genetic variation in offspring. All right, meiosis. At this point, we are looking at gamete formation. So meiosis where are we? There we go. This is number 7. The result of the offspring will receive a better suited to the environment. Where are we? This is number 8. My bad. Variation, these variations cause leads to adaptation and change. These variations cause some of the offspring to be better suited to the environment. Um, so this means that they are more likely to survive to a reproductive age. And if they survive to the reproductive age, they are more likely to pass these variations down to their offspring. Ladies and gentlemen, these tiny, tiny, itty bitty variations over time is how evolution happens. For example, myself, um, I only had one set of wisdom teeth. Like when you turn around, like, I don't even know how old I was. I was in high school still. And you have to go to the dentist and they take out your wisdom teeth and they normally have tops and bottoms. For some reason, I only had bottoms. So potentially that gene was passed on to my children. In fact, it was passed on to at least one of my kids because my son didn't have bottom wisdom teeth. I'm sorry, he didn't have tops wisdom teeth either. Favorable variations sometimes get passed along, and that happens because of that crossing over stuff. All right. We are at number 9. If the result of crossing over creates offspring to be less suited for the environment, it might not survive. And that's just the reality of biology, of life on this planet. If an organism does not survive, it may not be able to actually reproduce. Like if it doesn't survive to reproduction age, it's not going to have offspring, which means it might not be able to secure a mate or maybe just doesn't survive at all. These unfavorable genes are not likely to get passed on to your offspring. One very, very simple thing that I would like to point out is how people are getting taller. As a general rule, that is for two reasons. Number one, our nutrition is getting better. And number two, more often than not, and I'm not saying in every situation, men prefer to have shorter women, and women prefer to have taller men. But again, and I'm talking in humans in general, in the American population, I'm not saying there's anything wrong with being tall or short, no matter which way you are who you are, and you are beautiful. But that being said, we do see this tallness favorability passing along easier in the American genome. And we as a country are getting taller and taller over the last probably 40 years or so, because for some reason tallness is seen as favorable in our culture. Again, over millions of years, hundreds of thousands of years, crossing over eventually leads to evolution. All right, that's number 10 on your note catcher. All right, so now we are at gamete formation. This is A, B, and C. Oops, nope, at least it's just A, B maybe. Meiosis produces 4 haploid cells that are different. Remember that crossing over creates a weird variation. So in males, meiosis can result in 4 sperm cells. So not only are they different from the parent cell, but they are different from each other. Each sperm cell down there on the bottom is 100% unique from each other, as well as the original parent cell. However, when it comes to females, 4 cells are produced, but only 1 is a functional egg. All of the cytoplasm and organelles are put into 1 egg, and the other 3, even though they exist, we know them to be polar bodies because they will never actually function as a true egg. All right, so this is important. Oops, here we go. Comparison of meiosis and mitosis. Because again, remember, we are looking at cell division. There are 2 different reasons why cell divide. Mitosis creates body cells. It creates growth. It creates healing and repair. Meiosis, on the other hand, is cell division only for the purpose of creating egg and sperm cells. Meiosis is only for the purpose of sexual reproduction. Mitosis, of course, growth, repair, and even asexual reproduction. Mitosis does those three things. Meiosis only occurs for the formation of egg and sperm cells. This is part A of the comparison. All right, moving on. Part B. So in meiosis, each diploid cell divides twice and produces 4 total cells. In mitosis, each diploid cell only divides once and makes 2 cells. Okay, so meiosis makes 4, mitosis makes 2. In meiosis, each of the 4 cells contain half of the number of chromosomes as the parent cell, but mitosis, each new cell has the same number. So remember, mitosis, which we talked about first last week, these make identical cells. They're all identical with the same number of chromosomes. Meiosis, however, we are making those haploid cells, which means we are going to only have half of the number of chromosomes as in the parent cell. So a few differences already. Meiosis divides twice and we make 4 cells. Mitosis divides 1 time and we get 2 cells, and those cells are identical as the parents. In mitosis, the homologous pairs do not come together to form tetrads. In meiosis, we do come together to make tetrads. And again, because those tetrads are forming during meiosis, we have crossing over that occurs. And of course, because there's no tetrad in mitosis, then there's no crossing over in mitosis, which is a good thing because remember, mitosis wants to make identical cells. Meiosis, on the other hand, makes me. We want to have some genetic variation. Finally, in mitosis, the 4 haploid cells contain different combinations of chromosomes from each other. In mitosis, the offspring, the resultant cells are identical. So again, the result of meiosis, right? 4 haploid cells, totally different from each other, completely different from the parent cell. In mitosis, the result is 2 daughter cells, which are identical to the parent cell. All right, ladies and gentlemen, so that is the end of the notes. The next thing that you need to do is my little exit ticket, which is a Google form. So good luck with that and peace out.